So good day everyone. So we are the group 5 and we were going to report about cellular respiration that would cover the aerobic respiration and anaerobic respiration, where we tackle here the 4 stages of aerobic respiration, which is the glycolysis, the acetyl coenzyme A, the Krebs cycle, and the ATC, or the electron transfer chain. So these stages occur within a cell, especially in animal cell or either a plant cell. No, so when we say cellular respiration, it is the process by which food in the form of sugar or glucose is transformed into energy within the cell. This energy is stored in ATP molecules, which then power all sorts of cellular processes, which include aerobic respiration that require oxygen and anaerobic respiration that does not require oxygen. So when we say anaerobic respiration is where there is no energy present. So ATP is created through other chemical substances that do not require oxygen. So however, these processes do not produce as much as ATP as aerobic respiration. While in aerobic respiration, it includes the 4 stages which we are going to tackle individually by our group. So the first stage is the glycolysis, in which the glucose molecules are broken down to create ATP in a cell cytoplasm, where pyruvate and NADH are the 2 substances produced, as well as the 4 ATP, while 2 ATP was consumed. So the next stage is the acetyl coenzyme and CO2 that was created using the pyruvate in glycolysis that occurs in the mitochondria. And the next stage is the Krebs cycle. So the acetyl coenzyme A was used to create more NADH, FADH2, CO2, and additional ATP molecules. While the final stage is the ATC, where NADH, FADH2 and the oxygen are used to create massive amount of ATP through the stage, so in which water molecules was also created in this process. So that was the topic that we were going to tackle individually that cover the cellular respiration. So for now, we were going to first tackle the first process of aerobic respiration, which is the glycolysis. So we need to remember that glycolysis is the breakdown of glucose and involve a lot of reactions. So what can you see here is the overall diagram of glycolysis. So when you say glycolysis, it provides a significant means to synthesize a small amount of ATP in the absence of oxygen, and also to generate aerobic acid that is essential intermediary metabolite. So in glycolysis, we were going to tackle 9 steps. No? So in the first step, as you can see here in the diagram, is it will involve activation of glucose, which is followed by oxidation reactions of the glucose fragments, the synthesis of ATP, and the formation of pyruvic acid. So in here, phosphorylation happen. When we say phosphorylation, it is the addition of phosphate group to either the reactants or the product. So in here, glucose is phosphorylated by means of an ATP acting with the enzyme hexokinase and will produce glucose 6-phosphate. So we need to remember that glucose is a type of sugar in which dili start ang metabolism and as a source of energy also. So in here, makita nato nga ang glucose is contains 6 carbon molecules with the use of ATP and with the help of enzyme hexokinase. So the product, we call it glucose 6-phosphate because the phosphate group from ATP, in which is adenosine triphosphate, meaning there are 3 phosphate group, but in the first step, our ATP here will transform to adenosine diphosphate, means it will have 2 phosphate group, and the other phosphate group was attached to glucose and make it glucose 6-phosphate. So merely, we need to remember that the step 1 is with the help of enzyme hexokinase, it enables our ATP to create to phosphorylate glucose. So that was happened in step 1. And for our step 2, so our glucose 6-phosphate is converted to its isomer, which is the fructose 6-phosphate by the enzyme phosphoglucoisomerase. So when we say isomer, it is two or more compounds with the same formula but having different arrangement. So our glucose 6-phosphate and fructose 6-phosphate is the only differ in arrangement of hydrogen and carbon, oxygen, but they only differ in arrangement. So the step 2 is merely the conversion of glucose 6-phosphate to fructose 6-phosphate, in which the enzyme is responsible, the enzyme responsible for this is the phosphoglucoisomerase. So that's it. And for our step 3, another ATP is spent in phosphorylating the first carbon of fructose 6-phosphate, which yields fructose 1,6-diphosphate. So meaning, our ATP phosphorylate to fructose 6-phosphate, but in here, the ATP will add another phosphate group to our first carbon. So in which having 2 phosphate group was called diphosphate. That's why from fructose 6-phosphate to fructose 1,6-diphosphate, in which having 1,6 is where the phosphate group was attached. No? So, muna siyang kakahitabo sa ato ang step 3. So, we're done with the step 3 and now we will proceed to the step 4 of glycolysis. So, in here, starting, it is indicated that starting of no energy has been released or no oxidation reduction occurred, and in fact, 2 ATP had been used and the molecules remain in the 6-carbon state. So, in which sa step 3, which is the fructose 1,6-diphosphate, was again na phosphorylate na po siya by adding phosphate group to the first and 6 carbon. So in this step, this 2 was split into 2 3-carbon fragments. So which is the dihydroxyacetone phosphate or the DHAP or the DHAP and glyceraldehyde 3-phosphate. So in this reaction, that they have same formula but different arrangement as an isomer. So and what happened is that the DHAP was converted to G3P, in which it is the end product of this step. So that was what happened here in the step 4 of glycolysis. So for our next step, so the effect of, which is the step 5 na ta, so the step 5 of glycolysis na ta. So for the next step, the effect of the splitting of fructose diphosphate is to double every subsequent reaction, because where there was once a single molecules, in which there are now 2 to be fed into the remaining pathways. So muna siya ang atong sa step 5 na dayon. So in this step, we generate energy in which G3P was involved in ATP synthesis, in which the NAD plus or the NAD plus that act as the coenzyme pick up hydrogen from G3P, forming NADH, an addition of organic phosphate, and produce diphosphoglyceric acid, in which there are 2 phosphate group. So in other words, in this step, glyceraldehyde 3-phosphate gikuhaan og oxygen sa NAD+, forming NADH. Ang katong NADH, mapaingon to siya sa which will go to the electron transfer chain. So ang ginuwangdan man to siya og organic phosphate, no? So makita nato sa diagram sa glycolysis, ang kanang PO4 -3 ba na dira sa taas, mo na siya atong ginatawag inorganic phosphate, and then mag-produce na siya og diphosphoglyceric acid because of the 2-phosphate group being added. So mo po na siya gakahitabo sa step 5 of glycolysis. For the next step, which is the step 6, so as indicated here, in aerobic organism, the NADH formed during step 5 will undergo further reactions in the electron transport system, where the final H acceptor will be the oxygen. In addition, 3 ATPs will be generated per NADH. So, in organisms that ferment glucose and anaerobically, the NADH will be oxidized back to NAD+, and the hydrogen acceptor will be an organic compound. So, what happened is that diphosphoglyceric acid will give its 1-phosphate group to ATP and to release ATP, in which because diphosphoglyceric acid is nakuha ng isa ka phosphate group, it will now become 3-phosphoglyceric acid, in which now the product for this for this step. So that means having 2-glyceraldehyde phosphate have 2 diphosglyceric acid and would mean there are 2 ATP produced here. So muna siyang gakitabo sa step 6. So we're done with the step 1, step 2, step 3, step 4, step 5, and step 6. And now we're going to proceed now in the step 7 and step 8. So what happened here in step 7 and step 8 is that 3-phosphoglyceric acid as the product of step 6. Step 6 will produce 2-phosphoglyceric acid. So in which phosphate group located in the 3rd carbon sa 3-phosphoglyceric acid is mobalihin sa ikaduha na carbon sa 2-phosphoglyceric acid. So, and from 2-phosphoglyceric acid will release H2O. It means na ay removal of water. So ang reaction nga ga-involved diri is ang condensation. So from 2-phosphoglyceric acid nakuhaan og 2 water molecule, now it will go to phosphoenol pyruvic acid, which is the product for this step. Again, the product for this step is the phosphoenol pyruvic acid. So, and for the last and the final step of reaction of glycolysis, in here, what will happen here is so the product of 7 and 8, which is the phosphoenol pyruvic acid, it will now donate its phosphate group to ADP. No? So ADP and will create ATP, which is ang nabili na lang dayon kay di-donate naman to niya ang phosphate group dito sa ATP, which para maka-create ng ATP, ang mabili na lang dayon is ang pyruvic acid. Remember that nag-donate ta diri og phosphate, which means nag-release ta og duha ka ATP, in which pyruvic acid will go to either TC cycle or the fermentation, depende na dayon sa organism nga gigamit. So muna siya ang nahitabo. That's what happened here in glycolysis that contain 9 steps. So and now we will going to proceed to the next process of cellular aerobic respiration in cellular respiration.
So we're going to discuss the steps in acetyl-CoA. As you can see in the figure, you can see the pyruvate. CO2 is going to rip off pyruvate as a waste product. You can see that we have 2 carbon molecule. When we rip the carbon dioxide, the energy is released and we use that energy to reduce NAD. Reduce this enzyme to make an NADH, and we know that NADH carries energy to the electron transport chain. We're going to continue to destabilize it. Coenzyme A is going to be added to acetyl-CoA, and coenzymes, when you add 2 molecules, they make them unstable. In the figure, you can see the acetyl group, and we're going to continue this process in Krebs cycle. We will be discussing about the Krebs cycle, or also known as nitricarboxylic acid. Krebs cycle is a series of events in which oxidation of acetic acid or its equivalent provides energy for storage in phosphate bonds or in a live organism. The first step is pyruvate. Pyruvate is derived through the glycolysis of glucose, which is a 6-carbon compound, and it is split into 2 molecules of pyruvate, which is a 3-carbon compound. In the next step includes the oxidation of pyruvate into acetyl-CoA by the enzyme pyruvate dehydrogenase complex, and in this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. The acetyl-CoA is a 2-carbon compound. As you can see in the figure, the next step is the acetyl-CoA combines with the oxaloacetate, which is a 4-carbon compound, to form citrate. Hence, resulting molecule is a 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase, and in this reaction, a molecule of NAD is reduced to NADH, and a molecule of carbon dioxide is generated. Hence, the alpha ketoglutarate is a 5-carbon compound, since the molecule of carbon is lost into carbon dioxide.
The first step begins in pyruvate. Pyruvate is right to the glycolysis of glucose, which is 6-carbon compound, then split to 2 molecules of pyruvate, which is 3-carbon compound. The next step is the oxidation of pyruvate to acetyl-CoA by the enzyme pyruvate dehydrogenase complex. And this reaction, a molecule of carbon dioxide and a molecule of NADH is generated. Acetyl-CoA has 2 carbon compound. The acetyl-CoA combines with oxaloacetate, which is 4 carbon compound, to form citrate. Citrate has 6 carbon compound. This reaction is catalyzed by the enzyme citrate synthase. Then the citrate becomes isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized to alpha ketoglutarate by the enzyme isocitrate dehydrogenase. In this reaction, a molecule of NAD to NADH, then a molecule of carbon dioxide is generated. The alpha ketoglutarate has 5 carbon compound, then converted to succinyl-CoA by the enzyme alpha ketoglutarate dehydrogenase. In this reaction, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is released. The succinyl-CoA has 4 carbon compound. The succinyl-CoA is converted to succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of JTP is generated. Then, succinate is converted to fumarate by the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2 is generated, which is used to the production of FADH2, then converted into malate by the enzyme fumarase. And last, the malate is converted to oxaloacetate by the enzyme malate dehydrogenase. And also, in this reaction, NAD is reduced to NADH. Thank you.
Hi, I'm Kaishi. Electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. What happens here is that electrons captured from the donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP synthase are the following complexes that are found in the ATC. In addition to these complexes, 2 mobile carriers are also involved, such as ubiquinone and the cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, ADP, and inorganic phosphate, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron one at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with the molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps create a gradient. The potential energy of this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Cyanobacteria, also known as the blue-green bacteria or the blue-green algae, and belong to the phylum Cyanophyta. It is a bacteria that obtain their energy through the process of photosynthesis. So the ability of cyanobacteria to perform oxygenic photosynthesis is thought to have converted the early reducing atmosphere into an oxidizing one, which dramatically changed the composition of life forms on Earth. By the process or by this simulating biodiversity and leading to the near extinction of the oxygen intolerant organism. So, according to the endosymbiotic theory, chloroplasts and plants and eukaryotic algae have evolved from the cyanobacterial ancestor via the process of endosymbiosis. So here is my diagram. It is the diagram of the photosynthesis happens in the cyanobacteria. So when the light here enter to the cell of cyanobacteria, the photosystem 2 execute the initial reaction of photosynthesis in higher plants, algae, and cyanobacteria. So it captures light from the sun to catalyze the transmembrane charge separation. So the pigment here in photosystem 2 is the chlorophyll, which the proton strikes from the photosystem 2 to initiate the photosynthesis. Then it will move to the photosystem 1, in which it obtains the replacement of electrons from the electron transport chain. So the energy that happens here is the ATP, and the NADPH provides the hydrogen atoms needed to drive the subsequent photosynthetic dark reaction. So, after that, the energy is being converted here in the process of photosynthetic process. So, the carbon is being assimilated or takes place in the cyanobacteria, and the oxygen is being released, global food chains, and the climate-neutral production of fuels, metabolites, and basic chemicals. So in general, green plants and other organisms uses photosynthesis because photosynthesis is the conversion of light energy into chemical energy, which is then used to power the cellular activity. Sugars which are formed from water, and carbon dioxide used to store chemical energy, with the oxygen produced as a byproduct.